another video of the HIV series where I'll talk about the pathogenesis and structure of HIV. So let's begin. Okay, today I have another set of questions for you. So let's begin with the questions. What are the NACO guidelines? So what are the NACO guidelines? The positive result of the first test, that is A1, is confirmed by A2 or A3. Okay, that is the first principle. What is the second one? The first test, that is A1, it is highly sensitive, and the A2 or A3 is highly specific. Okay, this is the second principle. What is the third principle? The three tests A1, A2, A3 should be based on different antigens or different principles. Okay, that is the third test. Okay, now what is the fourth one? The fourth one is supplementary or confirmatory test should only be used when either A2 or A3 is indeterminate. Then we will go for a Western blot like technique for confirmation. Okay, what is the next question? The confirmatory test for HIV. Answer them for yourself. What is the third one? Why are immunochromatographic tests, rapid tests used for HIV? They are used for post-exposure prophylaxis. And why do we need them? In case of needle stick injuries. Where do we need them? In case of healthcare workers. So we need it for post-exposure prophylaxis in case of a needle stick injury. Okay, and what is the last question? Which mode has got the least risk of transmission in HIV? The sexual mode has got the lowest risk of transmission, which we always misinterpret maximum of the times. Okay, so these were the questions. Now let's begin with today's topic, that is the structure of HIV. We have already given an overview in the first lecture, but today we will know it in detail. Let's revise some facts. HIV is of two types, HIV1 and HIV2. It belongs to the retrovirus family and its genus is lentiform. Okay, now we will go for the details. Okay, here we can see the outer envelope. We all know that HIV is an enveloped virus and it has got a lipoproteinous covering. So this is the lipid part and this is the protein part, as we can see. Then we will go to the electron dense cone shaped core. This cone shaped core consists of three things. First of all, it consists of matrix protein P17. See, this is the matrix protein P17. Okay, then we have the major capsid protein, that is P24, that is detected by ELISA, the most important protein that is detected within the window period. We have got the nucleocapsid protein, that is either P7 or P9, and then we have got two identical copies of viral RNA in the core. See, we have two identical copies of viral RNA. Then we have got the three most important viral enzymes: protease, integrase and reverse transcriptase, which we remember by the mnemonic RIP. Here also we see the same structure. The outer spherical envelope, which is lipoproteinous, this is the lipid part, and this is the protein part, which has got a knob-like structure and a pin-like structure. Then inside it we have got, see, this is the envelope written here, and it has got the surface proteins, which I will come, and then we have got the inside the core, that is a icosahedral symmetry. And first of all, we've got the matrix. See, the matrix is made up of P17 protein. Then we have got the capsid, that is made up of P24 protein. Then see, this is the capsid. This thing is the capsid. Okay, then we have got the nucleocapsid, that is made up of P7, that is made up of actually P7 and P9. And then we have another protein as well, that is called the budding protein. This budding protein is the P6, that is also involved in the structure. Then we have got the two single stranded RNAs, or two exact same copies of RNA. See, this is the two RNA, and then we have got the three enzymes. First we have got the reverse transcriptase. See, this is the reverse transcriptase. Then we have got the protease, this is the protease, and then we have got the integrase. So this is the integrase. So we are clear with the structure. Now we will come to its genome. Genome will tell us a lot about the structure. See, first of all, we got this LTR and LTR. We will begin with the LTR gene. This is called the long terminal repeat. This long terminal repeat, what does it do? It flanks the HIV RNA genome. Means it holds the HIV RNA genome with its two hands. Okay, and these are special ends also. This LTR is a special ends by which it attaches to our DNA. Okay, to our host DNA, this LTR attaches, and then it can divide with the help of the host machinery, that is with the help of us. तो हम LTR को ऐसे सोच सकते हैं कि there are two security guards. Okay, these are the two security guards. This is one LTR and this is another LTR. Okay, this is one LTR and this is another LTR. Now these two security guards are holding a rope in their hand, and this rope is the genome. Okay, now when these two security guards get to see the host DNA, that is our host machinery DNA, it allows, or it comes and attaches to this host DNA, and then it starts replicating. But it does not allow any random person to enter it. If it sees the host DNA, this is our host DNA, only if it sees that, it multiplies. Okay, so see, this is also the structure which we can see here. This HIV genome. First, see, this is the LTR region and this is the LTR region. This is the 5' LTR and this is the 3' LTR. We will again get into that. First, let us look into the other genes. And this LTR gene is also required for the initiation of transcription. Okay, this contains control regions that bind host transcription factors NF-kappa B, NF-AT, SP1 and TBP. So these transcription factors of the host will help in the division of the HIV RNA or multiplication of the HIV virus. So this NF-kappa B is the gene that is an inflammatory mediator gene. It secretes cytokines, or it helps in the production of cytokines or other inflammatory mediators like chemokines. Okay, now let's discuss a very interesting point, that is RNA contains the LTR region contains the RNA transacting response element that binds TAT. Now what does it mean? Okay, let's look into the diagram. Here we see that there is a lock, and this lock stands for the TAR. So the TAR is the lock, and here we have the TAT region. So this is the TAT region. This TAT goes and binds to the lock region. This TAT acts as a key which goes and binds to the lock region, and there is unlocking and there is transcription. There is transcription of HIV genome. Okay, now what happens is this TAR region is the one which helps in the active transcription of the HIV RNA virus, because once the transcription starts, it stops very soon. It takes very little time to stop. So when this TAT binds to the TAR region, the transcription process continues for long. Okay, so the lock and key theory can also be applied here. Now we will go towards the GAG gene. See, here in the GAG gene, we can see here, and we can also see here, the GAG gene consists of three regions: the MA, the CA and the NC. Basically, we can think it as a whole gene segment. This whole gene segment can be divided into three parts. One codes for the matrix protein, one codes for the capsid protein, one codes for the nucleocapsid protein. Okay, so here we can also see this is the matrix protein P17, this is the capsid protein, this is the P24, and we also have the nucleocapsid protein, that is P7, and along with P7 and P9, along with that we also have the budding protein, that is P6. So this is our role of GAG gene. This is a very important gene to remember, this GAG gene, this POL gene, and along with that this ENV gene. But we will go one by one and look into the detail of every gene. But to remember, we should remember the GAG, the POL and the ENV, because it is important in lab diagnosis as well as in the treatment of HIV. See, we can also see here that is the GAG gene codes for the matrix protein, the capsid protein, the nucleocapsid protein, and also the P6. Next, we will look forward to the Pol gene. The Pol gene is basically the polymerase A that codes for our RIP enzymes that is present in the core, that stands for reverse transcriptase, integrase, and protease. Let's look into the diagram or the gene. See, this is the Pol gene. This is the Pol gene. This consists of the PR. This consists of the, that is a three in the enzymes it codes for. This is the Pol gene that codes for the protease, the integrase, and the reverse transcriptase. As we can see here, we can also see in the figure that I had drawn here. See, this is our Pol. This is our Pol gene, and this Pol gene codes for the reverse transcriptase, the integrase, and the protease. Okay, these are the three molecules we can see. So this is the role of Pol gene. This Pol gene is also very important to remember, as I already said. See, this Pol gene is very important to remember, the GAG and the Pol. Now let's move on to the next. Okay, next we'll go for the VIF gene. This VIF gene stands for virion infectivity factor. Virion infectivity factor. Okay, so let's learn what does it do. This VIF inhibits the ApoBec gene. What is a ApoBec gene? ApoBec. Now, what is ApoBec gene? This ApoBec gene is a cytidine deaminase that converts the cytidine base into uracil. This cytidine is converted into uracil, which means as DNA, now the HIV DNA is double stranded, right? So when it after reverse transcripting, so the HIV DNA is double stranded. So if we have C here, there used to be G. Now it's not C and neither G. Now it is replaced by U. Now this is replaced by U. So there, here it also will be replaced by A. So here from GC there will be a conversion into AT. Okay, or AU. Here we will have AU. So therefore what will happen is AU, there will be a over mutation, or there will be a mutation that will convert this C into U and G into, this G will be converted into A. Okay, now what will happen is the HIV RNA will be defective, so there will be no more multiplication. This HIV variants will no more multiply. Okay, so therefore there will be no more amplification, or there will be no more multiplication of the HIV RNA viruses. Now VIF comes to know this, the HIV knows this, so it goes and inhibits the ApoBec gene. Okay, so it is its function. So this HIV RNA is very clever. Okay, we have the VPR gene. What does the VPR gene do? The VPR gene helps in replication amplification. This helps in replication amplification. There will be many more or much more replication of HIV RNA. So here we had the TAT gene and the TAR region, which helped in transcription, and the VPR region helps in viral replication. We have the REV gene. This REV gene, what does it do? It helps in taking the viral RNA into the cytoplasm of the host. Okay, it takes the RNA into the host cytoplasm. Next we have the VPU gene. This VPU gene helps in the release of the viral particles. So a lot of virus particles is made, a lot of virus particles like these are made, and then it helps in its release. Okay, it helps in release out of the cell and infect other cells. Okay, next we have the most important gene here, that is the ENV. That is the ENV, that is the envelope, the codes for the surface proteins GP120 and the transmembrane protein GP41. This GP120 is the main receptor that helps in binding of the RNA virus with the host cell DNA, and this GP41 is a fusion protein. Okay, So, and another we also have is NEF. What is NEF? This NEF inhibits the MHC1 or the major histocompatibility complex 1, that basically suppresses our immune system, that basically suppresses the host's immune system. Okay, so these were the genes. Another, again we have the LTR. Now let's look at the points regarding it. See, here we have the VIF, and it destroys the apobec 3G, which is a cytidine deaminase, and you can make more research on it if you are interested, because it has got very interesting facts regarding it. How does it name derive and all, that is not required here. Next we have VPR. VPR amplifies replication. Okay, then we have the TAT gene that I'm talking a lot from the very beginning. That is a transactivator protein or transcriptional activator. What does it do? It enhances viral transcription by binding to TAR. What is TAR? Transacting response element. That is basically a piece of RNA or a hair loop shaped RNA. Okay, a hairpin shaped RNA that it binds to, and there is viral transcription. Next we have RAVE. What is RAVE? This REV gene takes the viral RNA to our cytoplasm, as we can already see here. Next we have the VPU, which increases the viral release. Next we have the ENV, which codes for GP120 and GP41, and together we call it the GP160 that I'm talking about in every video. And next we have the NAVE protein. Okay, I mean the NAVE gene. What does it do? This NAVE down regulates the MHC class 1 molecules and suppresses our immunity, or suppresses the host immunity. That's it. Okay, now we will begin with the life cycle of HIV and the pathogenesis of HIV. See, first of all we have the virus particle with the GP41 region and the GP120 region, or the GP41 protein and GP120 protein. First of all, GP120 is the main receptor that binds to the CD4 T cell receptor. It can also bind to the macrophage. So it is the cell, this is the CD4 cell, this is the whole is a CD4 cell, and this has got the receptor. First of all it binds to the CD4 receptor. See, the first step, GP120 CD4 binding, it binds. And now what happens, this shows is real आइडेंटिटी, मतलब असली चेहरा अब निकल जाता है. सो इट लीड्स टू द कन्फर्मेशनल चेंज. इसका एक हाथ था जिस विद द हेल्प ऑफ विच इट हैड बाइंडेड टू द CD4 receptor. Now it has got two more hands because now it has undergone a conformational change, and now GP120 has two more active sites. See, it has two more हाथ, and it binds to the co-receptor. The co-receptor is either CCR5 in case of CD4 cell. So here with the as we are dealing with the CD4 cell, here it will be a CCR5 co-receptor, and if we were dealing with macrophages, it would have been a CXCR4 receptor. See, it is binding to CCR5. It has bind to the main receptor of CD4 receptor. Now it has binding to the two co-receptors. That is, one is with his two hands, the CCR5. That is the second step. Now we will move forward to the third step. Now GP120 was the main receptor. Now what is the binding protein or the fusion protein? This is GP41. तो असली काम किसका है? GP41 का. GP41 helps in the release of the core of the viral code into the host cell, that is into the CD4 cell. See, it has released its enzymes. It has that is the RNA genome along with its enzymes, or also the capsid and the matrix protein. So it releases its reverse transcriptase enzyme, integrase enzyme, and all of that along with the HIV RNA genome. So first of all, what happens? This RNA is released. This two single-stranded identical copies of RNA is released. Then they undergo reverse transcription, then a RNA DNA hybrid is formed, and some intermediate steps takes place, and finally this HIV DNA is formed. I will tell the intermediate steps in the pathogenesis in even more detail. Now let's look at the overview. So this is the HIV DNA. HIV DNA is formed. Now it migrates to the nucleus. Not only the HIV DNA along with its matrix, along with its capsid, all of them enters into the nucleus, and this we call as the pre-integration complex. This whole thing of matrix and capsid is called the pre-integration complex. And after that, when it with the help of the integrase enzyme, see this integration of provirus into the host cell genome, with the help of integrase enzyme, it binds the host DNA. So it is now called a provirus. An important thing to remember, that now it is called a provirus. So what happens? With the help of integrase enzyme, the HIV genome binds to the the host DNA. Now what does it undergo? It further undergoes transcription and translation, and what happens? It's synthesis of HIV proteins and assembly of virion core and structures, and this the lipid part of the envelope is synthesized from the host machinery, and then the HIV core plus the envelope is together formed and released with the out of the cell with the help of what? With the help of REV, that is REV genome, or the REV gene that helps in the release of the mature virion. Now new HIV virion is formed, which goes and infects the other cells. In the last part of the video, that is the actual pathogenesis or the flow chart. We have understood it in detail. Now let's make a flow chart, and so that we can write it for the exam. This is also a very important long question for the exam. See, first of all, what happens? HIV binds via GP120 to CD4 receptor on target cell, that is the T cells or the macrophages. Already I have shown in the diagram and or the figure. And then HIV through GP120 also binds to the core receptor, that is CXCR4 T cells in case of T cells and CCR5 in case of macrophages. So we can see what I was talking about is binding of the GP120 to the CCR5 or the CXCR4. We can see it in the diagram. Now let's move on. Okay, next, GP41 facilitates the fusion of HIV to the target cell membrane of the host cell. That I have already shown. This is the fusion protein GP41. This GP41 is the fusion protein. Next, what happens? Nucleocapsid enters into the cell cytoplasm. I have already shown that is a nucleocapsid along with the core. The core of the HIV is entering into the cell cytoplasm. Then reverse transcriptase synthesizes single-stranded DNA from the two single-stranded RNA strands, that is the two identical copies of RNA. Then what happens? RNA DNA hybrid is formed. After RNA DNA formation, hybrid formation, this RNA part is not required anymore. This RNA is degraded by viral endonuclease. Next, what happens? Single-stranded DNA replicates from double-stranded DNA using the host cell machinery. Okay, so single-stranded DNA is now converted into double-stranded DNA. Not the host cell machinery, or not only the host cell machinery, but also the reverse transcriptase enzyme. It uses both of it to form the double-stranded DNA. Now what happens? Now double-stranded DNA enters nucleus into the host cell, and that is called the pre-integration complex because it enters with the matrix and the other proteins. See, we can see here in the diagram. See, here we can see integration happens. Then first of all, this HIV DNA comes into the cytoplasm. This it comes into the cytoplasm with which gene? Look in the previous segments and answer yourself. Then integration of provirus takes place. Then this HIV genome formation takes place. Okay. Now let's look into the rest of it. This is integrated by integrase into the human genome, and then there is multiplication inside the cell and the increase in number to virus particles, and then the virus particles is released out with the help of another gene. Let's look at the previous segments of the video and you can answer it for yourself again. And this released out and infects the other cells, and then it goes into the latency period. The period of latency comes then, that is a period of 10 years. We have discussed it in the previous video. You can check it out, and that was the whole pathogenesis of the HIV virus. And I think this is the most interesting part, the life cycle of HIV in the infection along with the pathogenesis. We also learned for the HIV genome and the HIV mature virion. So that was it today. Thank you.